Hello. Good afternoon, everyone. Uh, my name is Ping Wang, and I am uh, from Department National Special Laboratory and the Department of Biomedical Engineering and College of Biomedical Engineering and Instrument Science, Zhejiang University, Hangzhou in China. The title of my talk is Bioelectronic Nodes and Bioelectronic Tongue in vitro and in vivo. Firstly, I briefly introduce our university, Zhejiang University. This is uh, our Yuquan campus, which is one of many campuses of Zhejiang University that focus in engineering and information. And this building is our college building. Our college is biomedical engineering and instrument science, and this building is also our lab and department. The contents of my talk include the five parts. One is introduction, and the second one is electronic nodes and electronic tongue. And the third one is electronic nodes and electronic tongue. Fourth part is application for bio-nodes and bio-tongue. And the final part is conclusion and future work. Introduction. Uh, as we know, uh, the two of basic sense of human beings, olfaction and gustation, play a very important role in daily life. So biomimetic electronic nodes and electronic tongue are very important for recognition environment conditions. The physiological structure of human smell and taste is shown in figure. The smell is first contracted by the olfactory epithelium. After olfactory epithelium cell response, the response signal is transmitted to olfactory bulb, which proceed by uh, amplification and filtering signal, and finally to olfactory contacts. Taste has a similar process, but taste substances in the solution first contact with taste cells here, which respond to the signal eventually to the brain stem and uh, brain stem and then to the taste perception context, uh, producing the image of taste. Uh, Although the details of smell and taste are not well understood, but engineering researchers have long been designing and developing artificial instrument, that is, electronic nodes and electronic tongue. We hope they can work like a human noses and tongue. So the second part is about electronic nodes and electronic tongue. Electronic nodes, the term was presented by in the 1980s. It became apparent that the animal and the human olfactory system operates on the same principle. That is mean relatively small number of non-selective responses receptors can determine thousands of different orders. So electronic nodes is a kind of electronic or optical analysis instrument, and well, it should be a kind of bionic instrument. So in the world, at present, structure of electronic nodes is shown in this figure. This is measuring order, and firstly contact with chemical vapor sensor array, and then through recognition algorithm. At present, mostly use some intelligent algorithm like artificial neural network to finally give recognize results to different order and different food and so on. Electronic tongue, that is e-tongue. This term was presented in 1990s. It became apparent that animal and human taste operating on the same principle. So electronic tongue is a kind of electronic or optical analysis instrument, and well should be kind of bionic instrument. This photo come from Russia, Petersburg University, and Andrei, Lake Andrei, and Dima laboratory. This electronic tongue is also have commercial products in the world, and this instrument is famous Professor Toko from Japan University, and his taste sensor consists of biomimetic memory, similar memory of taste cell. So at present, Enos and Etan have many applications in the world. For example, in environmental toxins and pollution monitoring, and medicine body function monitoring, and food processing monitoring, and industry process monitoring, and so on. We also, especially in my group, research Enos and Etan many years. Is also a very early group in China and in the world. We developed several nodes to diagnose lung disease like this. we firstly using GC-mass to determine the biomarks in the breast human breast, and then we developed the inodes to related genomes GC-mass to uh, sampling breath gas and using uh, uh, intelligent algorithm to diagnosis the different disease like uh, patients and healthy people. And this is our inodes for disease diagnosis like this. This is very small inodes. This is larger inodes. And we also make many clinical experiments in hospital. At present, we can screening the different people from hygiene and lung cancer and disease and so on. This is our laboratory developed the inodes for water monitoring, and this is we developed and fabricated the different micro ion sensor array to detect the different heavy metals in water. And we also put our ethane in SIP to monitor the water pollution, especially sea water pollution. This this SIP can run real time every day. The third part is about bioelectronic nodes and bioelectronic tongue. Bioelectronic nodes and bioelectronic tongue. The biggest difference between bioinodes and bioetang and early inodes and etang is that biosensitive materials such as smell and taste receptors, cells, tissues, and organoids are used as sensitive elements. So its response characteristics are more similar to biological smell and taste senses. The detection ability of conventional inodes and ethane mainly depends on absorbability or catalysis of those sensitive materials to special odorants and tastings. Although some achievements uh, have been made, this method still has limitation in sensitivity and specificity compared with the biology bending of specific odorants and tastings to the olfactory and gustatory receptors cells. So, and electronic tongue uh, have the similar characteristics and advantages. At present, um, besides uh, besides bioelectronic tongue also combined with cell culture and macro fabrication technologies. We incorporated living olfactory and taste cells or tissues as sensitive element with neuron chips in vitro for biomimetic olfactory-based biosensors. So here, in Bioenergy and Bioelectronic, we using uh, smell and taste cells as sensitive elements, and combined with the transducer um, from the uh, from the uh, from the sensor. Uh, Here we also developed the bio nodes and olfactory neuron network, including olfactory epithelum and olfactory bulb. This is our developed the bioenodes chip. Here we culture OE and OB on the surface of a chip. And we also developed the olfactory and gustatory tissue and organoid as sensitive elements. This is our bioinosabeta tongue with smell and taste cells. Here we using receptors based bioinodes and combine transducer from the receptor based bioinodes. Uh, this transducer is a surface acoustic wave because this transducer have very high sensitive uh, to to uh, to measure the odors, and this device uh, can use micro nano fabrication process, so it uh, can be uh, produce produced many macro uh, devices. These are our bioinodes and bioetan with living cells and tissue and organoids. This is our tissue-based bioinodes. And this research work and research results have published in Biosensor Journal of Biosensor and Bioelectronics. This is our developed the organoid-based bioinodes, and our research results is in manuscript, and you can see talk of Nanjiang is also in this symposium. And this is our developed the organoid-based bioethanol. Here we use um, uh, test tissue from rat to culture the surface of device. And this is we developed in vivo bioinodes, and we have published some papers in international journals. For example, in biosensor and in bioelectronics and sensors and activators. And this is our in vivo bioethanol, and his some results also published in international journals like BB and Sensor B. We further purpose study is on bionodes and biotongue is we hope combine in vitro with in vivo to build the more more sensitive and specific bioinodes and bioethanol. The fourth part is the application of bioinodes and bioethanol. This is our at present research bioinodes for evaluation of odor masking. This inode is constructed by integrating mammalian olfactory epithelium with microelectrode array chip to read out the olfactory information as electrical signal from multiple tissue sites. The intrinsic odor response of olfactory epithelium is found to be represented by widespread sufficient temporal or oscillatory activity. The masking efficiency of fragrance is quantified by calculating the relative difference between the malodor and the binary mixture. Malodor plus fragrance response pattern results indicated that masking efficiency of fragrance is concentration dependent. This study demonstrates for the first time that capitalism on the biological sense of smell to create a biohybrid system provides an effective technique to resolve more complex biosensing related issues, such as odor interaction in mixture, as so in this figure. Under the application, the second example of application of bioenzyme bioenzymes for detection of other receptor interaction. Compared to conventional gas sensor or electronic nodes, one of the advantages of bioelectronic nodes is the ability to detect other receptor interactions. Previous studies in the bioelectronic nodes mainly uh, enforces the specificity and sensitivity detection of substances. However, the detection of inhibition is instructive for food or fragrance companies. Based on the previous research of our lab, we developed an in vivo bioenergy for assessment of masking efficiency of intravaleric acid. In future, high throughput measured for evaluation of masking efficiency should be investigated uh, to achieve more efficiently detecting other masking as shown in this figure. The third sample of application of bioethan for bitter and umami detection. In this talk, bioethan is developed for bitter and umami detection, utilizing red cardiomyocytes as a primary taste sensing element and macroelectrode array as a secondary transducer for the first time. The cardiomyocytes-based bioethan proposed in this talk provides a new approach for construction of bioethan and has promising application in taste detection and pharmaceutical study, as shown in this figure. The next example of application is tissue-based bioethan. This detection of gustatory stimuli depends on the activation of taste receptor cells. Taste perception occurs when taste compounds in the mouth contact the epithelial cells of the taste buds within the oral cavity. Then the taste information was transmitted to gustatory different nerves, as shown in this figure. The multi-channel result demonstrated that taste buds released the spontaneous signals simultaneously and displayed different responses to different taste stimulations. This bioethan can be used to recognize different taste substances in a way close to human sensory evaluation objectively and quantitatively. Final part is conclusion and future work. Bioelectronic nodes and bioetan in vivo and in vitro is one kind of bio-spared sensors system or intelligent instrument combined electronic sensors with biological olfactory and gustatory organ and pattern recognition techniques and so on. In this talk, we presented some novel ideas in series including culturing living olfactory and gustatory tissue and cells and cell neural network on the surface of devices, and implanted electrode in olfactory bulb and gustatory contacts. We future research work, including the several parts. One is AD disease detection and drug screening. AD disease detection and drug screening using our bioenos. We at present we have established the olfactory bulb neuronal network chip based biosensor of Dysmear model in AD. OB neurons were planted on the surface of MEA chip so that we can detect the extracellular signals in time after ABOS inductions. Next one is in vivo bioenos for odor masking evaluation. We continue research this field. And we also will develop the animal robot based on bioenos and bioetan in vivo. And also robots with olfaction and taste. This is our published books in China and in American and in American articles company and in Spring company in Germany. Finally, thank you for your attention. And if you have questions, you can contact with me and with our members in my groups. My email given in following. Thank you once again.